You know those crime shows where the villain thinks they've cleaned up every single trace? The crime scene looks absolutely perfect. But what if I told you that even the most invisible clues can tell an incredibly vivid story? Let's dive in.
So yeah, that's the big question, right? When the scene is wiped down, bleached, scrubbed clean, where do you even start? Well, the answer is pretty fascinating, and it all comes down to a science that makes the invisible suddenly visible.
Just imagine it for a second. Investigators show up, and the place looks spotless. To the naked eye, there is absolutely nothing. No weapon, no stray fibers, no signs of a struggle. It feels like a total dead end. But this is exactly where forensic science really gets to shine.
That's right. The most important evidence isn't about what was left behind. It's about what someone tried so hard to get rid of. The real key is in the microscopic traces that just cling to surfaces, seep between floorboards. And you know the most revealing of all these hidden traces? It's blood.
See, even when blood is wiped away, these tiny, invisible bits are left behind. And inside that blood is a very special component that basically acts like a homing beacon for forensic tests. It's the one secret ingredient you just can't scrub away completely.
So what even is blood? Well, about 55% of it is this liquid called plasma. But what we're really interested in is the other 45%. That's all the cellular stuff. This is where you find your red blood cells, white blood cells, platelets. And our target? It's hiding right inside those red blood cells. And here it is, the hero of our story, heme. It's a part of hemoglobin, the stuff that carries oxygen around your body. But for investigators, its most important job is that it acts as a catalyst. You can think of it like a chemical matchmaker. It gets reactions started and makes them happen fast. And that's the weakness they can exploit.
So how do scientists actually use this catalytic power of heme? Well, they've got a whole toolkit of special chemical tests. We call them presumptive tests, and they're designed specifically to react with it. Let's take a look at the three most common ones.
Okay, I know that sounds super complex, but here's what it really means in simple terms. Heme is a trigger. When you add the right chemicals, heme kicks off a reaction that creates a huge visual signal. It could be a bright color, a flash of light, or even a glow. It's like setting off a flare to shout, "Hey, blood was here."
First up is the Kastle-Meyer test. An investigator will swab a suspicious area, and then they add a couple of things. First is a reagent called phenolphthalein. The second is a simple oxidant, just hydrogen peroxide. Now, if heme is in that sample, it's going to trigger a reaction between them. And the results?
You get an immediate, vibrant flash of pink. It's a classic test. Quick, effective, and gives you a positive sign for blood right there at the scene. It's just beautiful chemistry in action, revealing what was totally hidden a second before.
All right, let's move on to the next tool in the kit, the leukomolecular green test, or LMG for short. The procedure here is really similar. You swab the area, you add the LNG chemical, and then you add that same oxidant. Once again, we're basically asking heme to show itself with that same catalytic reaction.
Only this time, the result isn't pink. Instead, the sample turns this really striking blue-green color. It's just another powerful way of confirming that heme, and therefore probably blood, is on that surface.
But of course, the most dramatic test, the one you've definitely seen on TV, is luminol. With this one, the chemical is usually sprayed over a much wider area, like an entire floor or a wall. But for this reaction to really pop, you need one more crucial thing: complete and total darkness.
And then you see it. This ghostly, kind of eerie blue glow appears wherever there are traces of blood. This is called chemiluminescence. Luminol is so incredibly sensitive, it can reveal huge patterns of cleaned up blood, showing the full hidden scale of what happened at a crime scene.
So, there you have it. A quick look at the toolkit. You've got 3 different tests and 3 different signals. From the fast spot check of Kastle-Meyer's pink to the wide area search revealed by luminol's famous blue glow, each one uses the power of heme to make the unseen seen.
So finding the blood is a massive breakthrough, obviously, but it's really just the first step. These presumptive tests tell you that you probably have blood. The real question is, what kind of story can that tiny, invisible trace actually tell us?
To really get why this is so powerful, just think about this for a second. Blood makes up about 8% of our total body weight. So that means even a microscopic trace isn't just a random clue. It's a significant piece of a person that's been left behind, just waiting to be analyzed.
And this is where things get truly incredible. That single drop, the one revealed by a flash of pink or a blue glow, it's not just a clue. It's a treasure trove of information. It's really the first chapter of a whole forensic story.
See, once a presumptive test is positive, confirmatory tests are run back in the lab, and from there, forensic scientists can determine if it's definitely blood, if it's human or animal, and even identify the blood group. Further analysis can potentially reveal the sex, age, and even the race of the person it came from. All of that from a stain that was completely invisible just minutes before.
So the next time you think something's gone forever just because you can't see it, remember that chemical secret that's hidden in our own blood. It leaves us with a final, really fascinating question. If this much information can be pulled from a nearly invisible speck of blood, what other stories are just waiting to be told, hidden all around us, right in plain sight?